Hi, welcome to Neuroverse
a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on structure and function. And I want to ask the question whether structure determines function and if there's a relationship or if they're completely unrelated. And I will be focusing slightly more on the perspective of proteins and biochemistry.
Ooh, what a surprise.
Yes. But I will also mention a little bit about neuroscience.
And I think I'll have some things to say about the brain.
Yes. So, first, in terms of proteins, they're usually organized in a primary, secondary, tertiary, or quaternary structure. The primary structure is just a chain of amino acids, and the secondary structure is the assembly of the amino acids, which now is a polypeptide chain, and it gets folded into patterns. And these can be alpha helices or beta sheets. Alpha helices tend to look like a looped coil. And the tertiary structure is made up of the secondary structures, which make up the 3D shape of the protein. Now, in a really interesting experiment conducted by Advinson, he wanted to show that the structure and the assembly of the enzyme determined its function. In order to do this, he took this enzyme called bovine ribonuclease and broke its disulfide bonds. These bonds then spontaneously reformed, and the enzyme regained its enzymatic function as well as its 3D structure. And therefore, he was able to show that simply having the amino acid code is enough for the protein to not only know how to fold, but also be able to conduct its function, and how it's all coded by the primary amino acid sequence.
Yeah, that's so cool that the primary structure also determines the higher order structures.
Yeah, exactly.
And the function.
Yeah.
Which perhaps is the highest order of the structures.
Wow, yes. So this all seems very simple and neat in theory, but there are several other factors in biology that add some layer of complexity to this simple proposal. For example, there exists cofactors, which also play a role in the folding of the protein. There exists post-translational modification, which is after the protein has folded, it can suffer a methylation, which is the addition of a methyl group, or a phosphorylation or acetylation, or even proteolysis, which is breaking down the protein. So
right. And that changes its function.
It changes its structure, and therefore it changes its function. And it puts into question this simple premise that the primary structure is enough to code for the tertiary structure, the whole structure of the protein, and therefore the function. If the protein was sufficient as it is, why does it often undergo all of these additional modifications after it's been folded? Another really cool aspect of this folding dilemma is chaperones.
Yeah. Which you mentioned in the enzyme-directed evolution episode.
Yes. Exactly. Chaperones are these really cool structures which help fold a protein into its shape. So oftentimes folded or partially folded or even completely unfolded proteins get threaded into the chaperone, and then due to the hydrophobic or hydrophilic properties inside the chaperone, it can bind to the correct amino acids in the protein and conformationally change the protein, so get it to fold into the correct structure.
What I find most interesting about this is that the structure of the protein, the end product, is determined by the dynamic
Yeah.
between its own composition and the composition of the chaperone.
Yes, it's
Yeah.
so clever.
Because it adds a level of another like dimension of a way to diversify, I think.
Mm-hmm.
Because then the same chaperone can create many different shapes out of the different proteins.
Yeah, exactly. So the same chaperone, depending on what type of chaperone it is, can be used for different proteins. It's not necessarily protein specific, which is really cool. It just goes to show, like you said, the importance of the dynamic interplay between the unfolded or partially folded protein and the chaperone. And what's even cooler is there's this chaperone that I studied in my undergrad called GROEL and GROES. And
Ooh.
yes,
That sounds really fancy.
it just means it's composed of two sections, a smaller section and a larger section. So GROES is smaller and GROEL is larger. And then there's like a cage on top, which is where the protein gets threaded, and then once it gets folded, it can then pass on to the second structure, the second cage of the chaperone, and then if it needs to get folded again, it gets passed back up into the first structure, and it keeps doing that until it's fully folded, and then in the end the cage like unlocks and it opens and it allows the protein to leave, and all of this is done through this dynamic of amino acids of the protein versus the chaperones, and it's all done just through hydrophobicity and hydrophilicity and the tensions between these two different components.
Yeah. I just have to say that I wish anyone who's listening to this could have seen Carolina's hand movements then. All the cage unlocking and folding.
I feel like biochemistry is so visual, and
Yeah.
sometimes it's difficult to convey it in a way that verbally people can visualize it. Yeah, if you're curious, I would definitely recommend you look up chaperone and just see the beautiful structures that come with it.
Yeah.
So, yeah, following this train of thought of unfolded versus folded proteins, a third of the proteins in our system are unfolded, which seems like an enormous amount.
Wow.
I know. And
Who would have thought?
Who would have thought? And you may ask, what is the function of these proteins? It would almost seem intuitive that proteins, like I've mentioned earlier, fold spontaneously by themselves. It's often energetically more favorable to be folded, but we do need disordered proteins because they carry out functions that we need their conformational flexibility properties in order to, for example, carry out transcription, because RNA polymerase 2 carries out transcription by being initially disordered, and then once they bind, they reorder, and this allows for transcription to occur, and we need them to be this flexible, and it's so neat.
Yeah, that's really cool.
Yeah.
Yeah.
And so that's an example. We also need unstructured proteins for phase separation, so forming separate compartments like the nucleus.
So it's again back to the idea of the structure being determined by the dynamic between two structures.
Yes,
Yeah.
exactly.
I was trying to relate this to the brain and thinking about neurodevelopment and the development of the structure of a single neuron is shaped by both intrinsic factors like the genetic makeup of the neuron, but also like extrinsic factors like cues, cell signaling, and the connections that it forms with other neurons, which promote, like when a connection forms with another neuron, it promotes the growth of that neuron. And so again, it's the structure being determined by the dynamics.
Yeah, it's so cool.
I love that
Okay,
idea.
so this adds a potential avenue that we could explore with the structure-function relationship. Is it the structure that's necessary for the function, or is it simply the dynamic that comes from that structure?
Also, another question I wanted to ask is, if structure does determine function, then what determines structure. And I think the only way that it works, in my opinion, is function determines structure. Yeah.
It's a bit of a loop.
Yeah.
I don't know.
Well, if you think of...
Maybe dynamic determines structure.
Dynamic determines structure, which determines function, which determines dynamic.
Yes.
Yeah, we love circles. And
if you can think of something that takes us out of this loop, let us know.
Yeah, please let us know.
So going back into intrinsically disordered proteins, they are problematic though, because 79% of these proteins are usually associated with cancer, mainly because they're so reactive. And a good way of thinking about intrinsically disordered proteins is they just have like all of their amino acids out and exposed, and they can like react with everything. And you can think of it as like a dog running into a room and knocking everything over.
An extrovert.
Yeah, just something that reacts with everything,
yeah.
and makes a mess. But within that mess, it's kind of good sometimes because it leads to variation, which is so important in biology, like we've spoken about.
Yeah.
And something which is so interesting is that proteins do have order promoting and disorder promoting residues. So they have specific amino acid sequences attached that kind of indicate whether or not the protein will be folded or unfolded, which I think is fascinating.
Yeah. Wait, so specific amino acids determines this?
Yeah. So the order promoting residues, I'm going to spell it out, but it might be a bit long. It's CWYFILHVNM.
Oh, so a specific sequence of amino acids.
Yes.
Okay.
And that's the sequence for promoting order. And the sequence for promoting disorder is KEQSPRGA. I know this may just seem random letters, but for the nerd in me, this is like really cool. How you can just like, these words, code, protein structure.
Yeah.
And it determines on whether or not the protein will react or not react.
Yeah.
So, yeah. Tying it back into neuroscience.
Yes.
Something really cool is that I was looking at the amygdala. Amygdala is usually a brain region associated with emotion and fear and pleasure and arousal.
Mm-hmm.
Yeah. And there's this protein called activity regulated cytoskeletal associated protein, which sounds like a mouthful. It's ARC. And ARC, exactly. And it's associated with plasticity in the brain.
Yeah.
There's been studies that show that the that the amygdala post-transcriptionally modifies this ARC protein, which, now this is me speculating. I'm speculating that maybe this has an effect on how memories are encoded in the brain. Because, so there's this protein that exists and it's related to plasticity, and then the BLA specifically, the amygdala specifically post-transcriptionally modifies it. So it changes the protein a little bit.
Well, yeah, actually, so synaptic plasticity is the ability for synaptic connections to change in their strength between neurons. And this is thought to underlie memory and learning.
Yeah.
And the mechanism of synaptic strengthening and weakening is all down to molecular mechanisms of like signaling cascades.
Yeah.
Which always includes the post-transcriptional modifications. Yeah.
Yeah. And so
in a way it's encoded in the way that protein structure it's encoded in protein structure.
Yeah.
Yeah.
Which is really cool.
Yeah.
And there's already a vast amount of proteins which each have like a very specific role and function coded by their very specifically arranged amino acids.
Mm-hmm.
And then on top of all this, there's these little post-transcriptional modifications, either adding a methyl or another functional group.
Yeah.
And this funnels and focuses down the function of this protein even more. And it's crazy how we need this level of detail and specificity for it to carry out its function.
Of behavior, learning and memory,
and memory, and just biology in general, and how it's so specific down to the molecule. And how you can scale it down to a molecule and then scale it all the way back up to the behavior.
So I was reflecting on this when I was thinking about the question, does structure determine function? Well, I cannot see why structure does not determine function. I'm ready to be convinced if somebody has an argument for this. But in terms of the brain, I was thinking about what determines a neuron's function. Well, first of all, it is its physical structure of what they call the dendritic tree, which is the part of the neuron that receives most of the inputs. But it depends on the channels that are contained in the membrane of neurons, because these determine its biophysical properties, which determines its ability to process signals and transform them into outputs. And this, therefore, the function in this sense is determined by the structure of the ion channels, because they have a structure in themselves. And another thing that determines neuron function is the activity that they receive, so the inputs they receive, which again is structural because it's the place that a neuron has within a neural circuit, within a pathway. And activity, as we've just said, can influence the strength of the synaptic connections, which has post-transcriptional modifications, but this can also alter the size of the physical size of the connections and the physical structure of the neuron. So for any neuroscientists out there, the size of the spines, the dendritic spines. And so, ultimately, this is all structure. And yet another thing at a more global network level is that the dynamics of a neural system also, in a way, has a structure. So this is more of an abstract structure. It's not necessarily a physical structure. But when we talk about network dynamics, we often explain it through a concept known as neural state space, which is the space describing the set of all possible neuronal states. So basically, each point in neural state space is a possible neural state in a moment in time. Within this exists something known as attractor networks. These are networks of neurons which are often recurrently connected, meaning they're connected to each other, and their activity settles to a stable pattern called an attractor. So an attractor is basically the set of states that the system tends towards in terms of activity. So an example of how this can be structural is there's something called grid cells, which are cells that fire in particular locations in space. Now, if you map the firing patterns of grid cells, the map that it creates, if pictured in 2D, in a 2D square, you can basically fold over, you can...
Like a sheet of paper.
How do I describe this? So imagine a sheet of paper. You can connect the two ends of the paper, the top and the bottom. So that makes a cylinder. And then you connect the other two open ends. That makes a donut.
Yes.
And this is called a torus attractor. And this is just one example of an attractor state, that can be visualized as a structure, and it carries out a specific function of knowing where we are in space. And different attractors are associated with different functions like memory or motor behavior, etc. So that's a really brief overview of attractor states. I think it's so fascinating how functions that emerge out of the brain structure can be mapped to a structure.
Yes.
That makes sense.
Yeah, that makes so much sense.
So
it's so fascinating.
It brings us back to the idea of like cyclical dynamics to structure to function.
Yes.
Wow.
I love our combined struggles of trying to visualize very like abstract and like concepts.
I've never tried to describe how to make a donut out of a sheet of paper.
And I've never had to explain what a chaperone cage looks like.
Well, we hope this episode stimulated your imagination to imagine some beautiful structures and how they can contribute to function.
Yes, and if you have an opinion on whether structure determines function, or if it's function that determines structure, or if it's dynamics, we'd love to hear your opinion.
Absolutely.